Greetings, and welcome to this series of vignettes that have to do with the ingredients of evolution. So, what we're going to do is we're going to consider here the fact of variation. What I'm going to try to argue is that variation is one of the key sort of clays out of which evolution is sculpted. And the way we'll do this is really by virtue of case studies. So the plan of action goes like this. Well, first of all, let me say that we don't really have to look very far to see the existence of natural variation. What I mean by that is, if you are on an airplane and when the flight arrives and everyone stands up in order to get off, you'll see very quickly the variation in human heights, and that's something that is well known and has changed over time in interesting ways, possibly due to diet and in human health and so on. But you can see here in this sort of double human histogram the distribution of heights of humans. So what I want to do is I want to basically use four distinct case studies. I want to start out by talking about viruses. That seems very pertinent given that we're doing this online. And after that, I'll say a few words about plants with special reference to Arabidopsis. Then we'll talk very briefly about flies, Drosophila in particular. And then finally, we'll talk about the finches that are well known from the Galapagos. So the fact of variation in viruses. I mean, this is a very, very interesting topic. And I think it's one that is obviously quite inspiring given the current moment that we're living in. I mean, we're at this point closing in on 2 full years. Well, we are at 2 full years from the moment when we all started hearing about the coronavirus in Wuhan. And so at the bottom, I show you two examples of so-called asymmetric viruses, one of which is HIV, another of which is influenza. And this is really super fascinating. And I would say that we're not really to the point where we understand very well what the, let me say, pathological or health consequences are of this variability. So there was this really great paper that was done in the early 2000s by Jordan Benjamin when he was a grad student at Caltech in the group of Grant Jensen. I've always really loved this paper because what they did is they explored the structure using cryo-electron microscopy of the HIV virion. And what you see is 3 categories. So at the top, the ones in red, you're getting 3 different perpendicular views, and it has this sort of characteristic ice cream cone shape. The orange ones are more cylindrical, and the bottom ones are irregular. And this is just to point out that there is this variability. Perhaps much more important, I'm going to move over to the side here because that orange part is the reason that I'm sitting in my room at home rather than talking to people in the classroom. That's the rise of the Omicron variant. So this is a very beautiful graph that comes from the NextStrain website, which I really recommend that everybody take a look at. And you can see that this goes back all the way to February or so of 2020. And what we see is basically the accumulation of variation here in the form of mutations in the genome. So in other words, there's different variants of the virus that get sequenced. There's 30,000 nucleotides. One can sequence them and then compare and ask, relative to the, let me call it the reference strain from Wuhan, how different is the currently propagating strain? So all of this is just to show you that in the context of viruses, this notion of variability is super well known. And HIV, SARS-CoV-2, influenza, the reason we get a new vaccination every year, all of that has to do with natural variation. So I would say, we're going to we already have made a little bit of a point about the idea that evolution is both a fact and a theory. And for the moment, I'm talking about the fact. The fact is that there is, in living organisms of the same species, there's variation. That's just a given. And let's take a look at a couple of examples from plants. So this is a very famous paper. You'll see the names of the authors here, Clausen, Keck, and Hesse, and we'll try to post this for you. I've always been fascinated by this as a case study because basically what they did is they looked at size and flowering and so on in a particular species of plant, and you can see in the map basically that we're looking at California, and we're basically from the left to the right going from west to east, and there's a change in altitude. And what the consequences of that are in a given species, that as you can see at the top, the nature of the plants varies. And so, there's all sorts of things of this variety. So I like this graph because it tells us about the change in flowering time and how that depends on drought conditions. And this famed study from Klaus and Keck and Hisi, as you can see, I'm just going to read this out loud. Here we see how the height of a yarrow plant depends on its genotype and the altitude at which it is raised, as shown by populations of yarrow plants grown in gardens at 3 sites that were at different altitudes, high, medium, and low elevation. For example, the green screen behind the plants of genotype 1 shows that these plants grow tall at high and low elevations, but are short at medium elevation, blah, blah, blah. So, that's fascinating. That's natural variation coupled to the environment in ways that, perhaps we'll have an opportunity to explore. Now, biology has been populated by all of these wonderful model organisms. And of course, in a way, the way that that an organism becomes a model organism is that it's useful for some reason, either that a community develops around it or there's some particular convenience that's associated with using it. For example, in the study of action potentials, well, people wanted to use a giant axon from squid because it was big, and that meant that they could put their electrodes in it in a convenient fashion. So Arabidopsis is a very, very well-studied plant. It's sort of like the E. coli of plants, and people have examined all sorts of features. You can see in the top that these phenotypic parameters are very different, and so there's all sorts of different versions of natural variation shown to you in this right-hand column. I especially like, for example, the response of these plants to metals, or to whether or not they, I think I have this here, whether or not they are performing either chemical or physical warfare, for example, by developing prickles, which are these so-called trichomes. But, for my purposes, the main thing that I want you to notice is that, as you can see in this column, that there's all sorts of different questions that one can pose about the variation in these different accessions of Arabidopsis. And, that's something that people have studied. And again, this is just to acquaint you with the idea of variation as the clay out of which evolution is sculpted. Okay, the fact of variation in animals. So speaking of model organisms with a great Caltech connection, Thomas Hunt Morgan was one of the founders of our biology department. He and his collaborators found these first mutants, for example, the white-eyed mutant that you see on the left-hand side, but there's a whole host of other mutants, and the mutants basically arise, I'm a mutant, so are you. I mean, we're all mutants in the sense that there's things about us that are different, and so in the context of flies, what they did is that they collected these mutants and then they bred with them, and used them in order to generate downstream versions in subsequent generations. And I just like this paper, which has to do with the diversity of female abdominal pigmentation. An apparently trivial point, but that said, something that one can actually go and quantify and score, if you like. So one can score the variation, and if there were, in some sense, if there were an advantage to this coloration, whether it was because of selection in terms of sexual selection, or in some ways because of camouflage or whatever, I'm making this up, but then that particular variation might be drawn from. There's variation in the number of bristles, and that's well characterized here, that I think people have tried to make these measurements. And when we talk about the forces of evolution, I'm going to talk about eye color. This is a case in which genetic drift, and when I say genetic drift, I mean something very simple, which is that the color of the eyes will depend basically on flipping coins, nothing more. There's no selection, it's just a matter of chance. So here we see that variation in bristle number is there for the taking. And I'm going to close with one of the greatest evolution studies ever done, and that has to do with the Galapagos, which hopefully, in light of the pandemic, it's not entirely clear, but hopefully we will be visiting in a couple months. And while there, for sure, we will see finches, and we will observe their beaks. So just a few comments. So, it's very worth trying to imagine Charles Darwin when he was your age. He left England at the age of 21 or so, if I remember correctly. He spent a 5-year voyage on a ship. There's all sorts of things to be said about that, some of which maybe I will say in a later vignette. But the main point is that, as you can see on the left of the map, off the west coast of South America, they spent 5 weeks in the Galapagos. It was very inspiring to Darwin. He didn't realize it at the time, unfortunately. So, there's a story there that has to do with whether or not he labeled which islands the finches came from and so on. But one thing that I would say is that, the finches have become a model for evolution, a deeply held and valued model. And in fact, David Lack, I would say more than Darwin, really pushed the finches as a key evolutionary story. For me, when I think about birds and Darwin, I actually think about pigeons, which is something I hope I'll come back to in a later vignette. So who are these finches? There's something on the order of between 13, 15 species of them. In his memoir, The Voyage of the Beagle, Darwin noted, almost as if in awe, one might really fancy that from an original paucity of birds in this archipelago, one species had been taken and modified for different ends. And we'll have similar things to say about the tortoises. And so there's all these different birds, and Peter and Rosemary Grant, over a long and very productive and insightful and creative and hardworking career, they basically studied carefully the variation in these birds. And they worked on an island called Daphne Major. If you look here in the middle, you'll see Santa Cruz Island. Up at the top where the airport is, you'll see Baltra. And just off to the left of this little airplane, if you look carefully, you'll see Daphne Major. And that's where they did their study. It's a little tiny island. You can see it here. And, if we're lucky, this is one of our evolution courses as we were arriving at Baltra. And you can see it's so exciting to look out the window and basically see this beautiful, mysterious island with such a rich history in the hands of the Grants. And so, what they did is they would go there and they would basically capture birds and measure them and mark them. And the reason that that's significant is that we will, in the next vignette, talk about heritability. Variation without heritability is not really actionable, if you like, from an evolutionary point of view. But given that it's heritable, then they could know which parents led to which offspring, and they could measure the correlation between the beaks, for example, in the parents and the beaks in their offspring. You can see also from this figure something that I've certainly been able to observe on my, say, 11 or 12 trips to the Galapagos, is depending upon what the weather has been like, what the time of year is, very much like California. I look out right now, and I look up to the mountains, and even in the former burn areas, it's quite green. But I just talked to the one of the guys on the fire road while I was riding my bike the other day, a fireman, and I was asking him his prospects for this season. He says, all this rain is going to lead to a lot of fuel. So, I can see this in the San Gabriels every year, and so can you. It's something that one can observe. Maybe the problem is that when we're used to seeing something every day, we fail to really look at it and say, I wonder what's going on in terms of variation. So how was the data collected? You can see in the top, that's Peter Grant on the left, and they would capture birds in nets, and he was an expert, and we will actually practice this. We have some birds and some calipers, and you can actually see how well you can measure the beaks and whether or not you believe they're error bars and so on. But the bottom line is that they, like Mendel before them, they measured characters. And one of those characters is the beak length and depth, and that raises all sorts of fascinating questions for me that are physics questions, which is how millimeter scale changes in beak depth can be related to the ability to crack open a seed. It's a really great physics question, I think, and I find it kind of surprising because it seems very thresholdish, meaning that maybe 1 or 2 millimeter change in beak depth is sufficient to lead to an ability versus not to crack a seed. So this is the essence of what I want to say. That's all. I just want to report that, here what the Grants are showing us is out of 751 birds, that there's this variation in beak depth. It's quite a wide variation. Interestingly, if you were to, I don't know if he published it in a paper, but I heard David Prober, who's a professor at Caltech who works on sleep, he made a distribution where the x-axis on his graph was number of hours of sleep per night. And it's funny, it ranges from like 2.5 to 14 hours or something like that, with a mean in around 7. And it looks kind of like this. So I'm just trying to say, what am I trying to say? That there is natural variation sitting there, which is the clay from which one can sculpt evolution. And there's island-to-island variation. Again, fascinating. Not something that you would have guessed necessarily on first go, especially when you look at Santa Cruz and Daphne, which are really next door neighbors. You will see I could swim from Daphne Major to Santa Cruz. So it's It's really not that big of a difference, but that's I think one of the wild lessons of modern evolution is both the rapidity with which it takes place and also the very, very short length scales over which it takes place. There's another place in Israel called Evolution Valley where opposite sides of the valley have sort of different evolutionary outcomes. So that's what I wanted to say about variation. It's an introduction to the idea. What I hope you took away from it is that we looked at all sorts of examples from the tree of life: viruses, plants, animals, flies, and finches. And what we saw is a great deal of variation. And it is amenable to systematic quantitative analysis. And in the next vignette, what we'll do is we'll talk about heritability, although you can see in this lower graph that on the x-axis we have the average depth of the parent's beak, and on the y-axis we have the average depth of the offspring's beak, and you can see this really nice and compelling correlation, which is essentially the proof, if you like, of the heritability. So with that, I stop.